In the first part of lesson 15C, we're going to do a simple regression. But in the second half of this lesson, question 2, we're going to turn the simple regression into a multiple regression using dummy variable. Now, in both of these parts of the lesson, we're going to look at how CO2 affects plant yields. Photosynthesis involves a light-driven chemical reaction between water and carbon dioxide in plants, producing sugars and releasing oxygen. CO2 enters plants through stomata in the leaves during the day, while respiration leads to oxygen intake and CO2 release. This process makes plants' net CO2 sinks due to the greater CO2 uptake during photosynthesis than is released during respiration. While factors like light, water, and temperature influence CO2 absorption, atmospheric CO2 levels play a dominant role. Now, variations in CO2 concentration stem from factors such as time of day, season, industrial emissions, composting, combustion, and neighboring vegetation and water bodies. In this experiment, we're going to look at this relationship between CO2 and crop yield using an experiment where we have northern greenhouses and we got southern greenhouses. And the conditions inside the northern greenhouses and the conditions inside the southern greenhouses we're going to hold them constant. So we don't really have to worry about composting, combustion, neighboring vegetation, and water bodies. We're controlling for the industrial emissions by having it in rural areas. We're controlling for time, day, and season by using northern and southern greenhouses. Why do we do that? Because northern climates will have, on average, shorter days and cooler winters, springs, and fall than southern locations. Southern locations, you're going to have a lot more sunlight spread out through the course of the year. The days are going to be generally longer than in northern parts of the United States. And the seasons are going to be longer in the south than they are in the north. So that's how we're going to control for that. Now, according to the figure 1 in this greenhouse carbon dioxide supplementation produced by the Oklahoma State University Extension, as CO2 levels rise inside a greenhouse, yield rises, peaks, and then falls. Peak growth occurs when greenhouse CO2 levels is between 1,000 and 1,200. So if you go straight up from 1,200 and straight up from 1,000, we see we're at the peak. And then beyond 1,200, plants are still doing better inside the greenhouse than outside the greenhouse. But once you get to this dashed line here, this dashed line represents plant growth being the same inside relative to outside. And that occurs at about 2,000 on the high end and about 300 on the low end. The data at the bottom of the table represents CO2 levels in various greenhouses your firm owns and the corresponding annual greenhouse yields. The middle column is CO2 levels inside the greenhouses, and yield is the yield. And we're growing the same crop inside the greenhouses. Every greenhouse has the same exact crops in it. We're trying to replicate the soil conditions. So we're going to control for a lot of things, but all we're going to do is we're going to vary CO2 levels. Now, the first 18 are greenhouses that are located in southern rural areas, while the rest of them are in northern climates. So we're going to copy this data to Excel. I'm going to grab the word greenhouse all the way down to the S in submit question so that I preserve the table structure when I copy it into Excel. Once I have Excel opened, I'm going to right click on cell A1. I'm going to click match destination formatting. Then I'm going to go to the very bottom and I'm going to get rid of this S. I'm going to use this data to estimate a simple regression. The Y variable is the crop yield. So I'm going to highlight that yellow. And then CO2 is going to be the X variable. I'll highlight that with light blue. I'm going to double check that my sample size is indeed 48. So I got 1 through 18 in southern climates, and I got 19 through 48 in southern climates. So I have 48 observations here. I have the first greenhouse, the second greenhouse, the third greenhouse. We're holding everything constant, but the CO2 levels. Maybe we haven't installed a thing that regulates CO2 in a very narrow range. Maybe the thing we're using is an antiquated monitoring system. And so we're going to use this opportunity to determine what our optimal level of CO2 is. Okay, so you want to click on data and then click data analysis and then find regression and then click OK. The Y range is the yield. So click on the yield word and then hold your left mouse button down until you get to the very bottom. You don't want any empty cells here. We're going from C1 to C49. And then click this button to the right of this box to load the X. So I'm going to grab the one in row 49 all the way up to CO2 levels. And I'm going to click labels because the first row are labels, the names of the columns of numbers. And I think that's it. I'm going to click OK. Here are my results. Let me format the decimal places for all these numbers that are not whole numbers, integers. Go home, number, and then I'll reduce the number of decimal places to 3 decimals. I'm going to make some changes here. I don't like the words that Excel uses. I'm going to change this to error. I'm going to change this to p-value. Most packages call this the p-value of f. And then I don't need this stuff here. Let me get rid of that. Most packages don't give you that stuff. And then let me just widen this out a little bit. Okay, so I need to insert the degrees of freedom. There's only one degree of freedom for the regression, and that's because there's only one X variable, CO2. There are 46 degrees of freedom for the error. I changed that word to error there. And there's 46 because there are 48 observations, but we're estimating two things. We're estimating the intercept of an equation and a slope of the equation. So the reason why this is 46, we got 48 observations minus the number of parameters we're estimating in the model, and we're estimating two. Again, the intercept and the slope. The total degrees of freedom are 47. The sum of squares due to regression, is this value. The sum of squares due to error, that's why I like to call this error instead of residual. The sum of squares due to error is that value. The sum of squares total is that value. I'm going to press enter to make sure I got the answers right. And I do. The mean square due to regression, notice that these two are equal. Well, they're equal because when we calculate the mean square, we're dividing the sum of squares due to regression by its degrees of freedom. And this divided by that is the mean square due to regression. Now, this is smaller than this number because to compute this number, we divided this number by the 46. We'd expect both of these values to be equal if CO2 was not a predictor of crop yield. If CO2 is highly insignificant, we expect these two values to be equal. But that's not the case here. Now, the F statistic is the ratio of these two. If I divide that by that, I get the F statistic. If this number is massive, if this number is close to 1, this T-stat is close to 0, this would be an insignificant, highly insignificant predictor of crop yield. And as a result, these two things would be equal, and this F-stat would be pretty close to 1. And the p-value would be close to 1. But that's not the case here, is it? The p-value, 0.041. Now notice the p-value of the F statistic and the p-value of CO2's test statistic are identical. This is not a coincidence. This is a result of this being simple regression. Because in simple regression, squaring the T-stat of the X variable gives the F statistic. And this is generally not true in multiple regression. I can see these two numbers being close to each other in a multiple regression of like, say, 10 X variables and one Y variable, where 9 of them have nothing to do with the Y variable. You might see something where the square of this is close to that. So generally in multiple regression, this is not going to be the square of that. But in simple regression, this will be the square of that. And that's why these p-values are identical. Again, it's not a coincidence. So the estimated intercept and the estimated slope each has a standard error. This divided by that produces this test statistic. This divided by that produces this test statistic. I pressed enter to make sure I got those right. Now, open up statdistributions.com to show you how to get these p-values. So I'm going to click on student T's, and then the degrees of freedom when you're looking for the p-values of the test statistics, you want to use the degrees of freedom of the error, and that's 46. I'm going to change this to 46. Now I'm going to enter the test statistic for the intercept, 6.318, and the p-value is 0. See that? 6.318 is way out here. So the p-value is 0. You can't see it, but this tail is shaded blue and this tail is shaded blue. You're going to see that when I change it to 2.104. When I change the t-value to 2.104, you're going to see two tails here. You got this area plus this area. And so when you add this area to that area, you get a p-value of 0.041, which is the same as what Excel gave you, right? Now, the way this works is we have two tails here because we're testing the hypothesis. The true slope coefficient, beta, is equal to 0. And this is the test statistic that results from performing that test. And this is the p-value. So our p-value is 0.000 for the intercept and 0.041 for the coefficient of CO2. Now the coefficient of determination, which is this value here, it's pretty small. It's 0.088. Now the way we get that, we take the sum of squares due to regression and we divide that by the sum of squares total. And it's not very large, is it? If I multiply this by 100, I get a percentage, 8.777. So the coefficient of determination is 0.088. If I multiply that by 100%, this implies that 8.8% of the variation in yield is explained by the model, and the model is a simple regression with CO2 and yield. This isn't very big. What's going on here? Well, the first regression, we're using all the data to see how CO2 affects crop yield. And we're going to run a second regression that uses only data from northern greenhouses. The first 18 greenhouses were located in the southern rural areas, while the others were located in northern areas. So what I'm going to do is I'm going to add a column here. I'm going to insert a column here. I'm going to call it D. We're going to have D equals 0 for the first 18. And then we're going to have D equal 1 for the rest. Where D equals 1, it's a northern greenhouse. Where D equals 0, it's a southern greenhouse. And this, notice it says N equal 30 here. How many greenhouses are in northern climates? How many rows do we have? Right there, that little white box to the bottom right of my cursor says 30R times 3C. Now we have 30 rows of data. So we have 30 northern greenhouses. We're going to rerun the regression with just these 30 northern greenhouses. I'm going to copy this 19 to 48. I'm going to copy it to here. I'm going to copy this column header. And then I'm going to click on data analysis, regression. Okay. I'm going to click on this button to the right of this box. I'm going to click on the yield all the way down to that last number. Click on this little box here. I'm going to reload the X box with CO2 and the last number. And I'm going to just click OK. I'm going to highlight all these numbers and format like I did before with 3 decimal places. I'm going to get rid of this stuff. The degrees of freedom are still 1 for the regression because we only have CO2. It's sum of squares due to regression. It's mean square. The degrees of freedom for residuals are 28 because we have fewer number of observations in this regression. The sum of squares due to regression. The mean square due to error. And then I'm going to go ahead. I'm going to put you on pause. I'm going to fill out this table and we'll come back. Okay, welcome back. I filled in the information. Again, we only have 28 degrees of freedom for the residual because now we only have 30 observations because we're only looking at the northern area greenhouses. Our F statistic is really large compared to what it was. It was only 4.426 before. Now it's 20.267. And the reason why that is because the mean square due to regression is a lot larger than the mean square due to residual. The sum of squares residual is very small here relative to the total. Why? Because the data is fitting better to this model. And that explains why this F statistic is really large and its p-value is close to 0. The slope for the 30 northern greenhouses is smaller than the slope when you do all 48 together. But look at the test statistic. The test statistic is only 2.104 when all the greenhouses are together. When you're only looking at the southern greenhouses, the test statistic is it's more than twice of what it was. And that results in a very small, tiny p-value. The coefficient of determination is 0.42 here. That implies 42% of the variation in yield is explained by the model. So we have to account for these differences that we observe with southern greenhouses and northern greenhouses. But there's a structural thing going on in our model. So simple regression on all 48 of the greenhouses is not sufficient. We need to account for that. So with the p-value really, really close to 0, that means this test statistic is really, really large, making the difference between this and 0 very significant. So we would conclude that the coefficient on CO2 is not equal to 0. We would reject that hypothesis. In my particular problem, the actual level of significance is 10%. In yours, it may be 5%, it may be 1%. It doesn't matter what that level is. What matters is how big the p-value is to the significance level. Now my test statistic is 4.502. The degrees of freedom have changed to 28. And you can see the p-value is 0 here. The area to the right of this value, underneath this blue distribution, you can't see it. And the area to the left of negative 4.502, you can't see it either. This test statistic of 4.502 is a long ways away from 0, which means the statistical evidence of this being statistically different than 0 is very large. So we're going to reject this hypothesis because, again, the p-value is smaller than the significance level. If your p-value is bigger than whatever your significance level is, then you're going to do the opposite of what I'm doing. If your p-value is smaller than your level of significance, then you're going to reject this hypothesis. Because what we're saying is this is big. It's bigger than 0 in a statistical sense. When we reject this hypothesis here, that means CO2 levels have a significant effect. Because it's positive, we say significant positive effect. I'm going to press enter to make sure that my answers are correct. The F statistic in the ANOVA table up here, this is the ANOVA table. The F statistic implies that the estimated model is significant. And again, we get that from here. The p-value is really close to 0. The F statistic is really large. And so our model is highly significant. The equation for the estimated model, I have CO2 here times this coefficient. And then we're going to add that to the intercept. We're going to use this equation to predict the 44th greenhouse's yield. So I'm going to do that in Excel. Make sure you're using the results from the regression of the 30 northern greenhouses. Okay, I'm going to find the data. Here's the 44th greenhouse. I'm going to paste it on the same sheet as my regression of the 30 northern greenhouses. I'm going to paste as values. I'll pull it down. This is the greenhouse. This is the CO2 level. This is that dummy variable. I can go ahead and delete that. This is the observed yield. I'll call it OBS yield. Then I'm going to do the predicted yield. To calculate the predicted yield, I take the intercept. I'm going to add to it the slope times the CO2 level that is in the 44th greenhouse. And that'll give me the predicted yield to 3 decimal places. Its CO2 level was 356. Now that is expected to result in a yield equal to what? That. If the production manager of that greenhouse increases CO2 by 1, what's going to happen? So what I'm going to do is I'm going to type equal, then I'm going to do plus 1. So CO2 went from 356 to 357. Then I'm going to drag this down. This is fine here. I want it 357, but I got to move this up to the intercept and I got to move this up to the slope. This is my model, my intercept plus the slope times the CO2 level. And the CO2 level happens to be in this purple box. If I increase the CO2 levels by 1 to 357, again, we're adding this to the product of the red box and the purple box. And then I'm going to calculate the difference here. The difference is 11,845. That's the value of the slope. The slope, remember what the slope is. It's the rise over the run. The rise is 11.845 in yield. The run is a 1 unit increase in CO2. That's how you interpret the slope. Down here, we have explicitly modeled the 1 unit increase in slope from 356 to 357. And that results in a predicted yield that is 11.845 higher when the CO2 is 357 relative to 356. And this difference is equal to the slope. The expected yield, or the predicted yield, when the CO2 is 357, the expected yield is going to be that value, right? The difference in these two yields is the value of the slope or the difference in those two, right? Either one works. Explain why the coefficient determination increased from regression 1 to regression 2. I think the best way to explain that is through a scatter plot, which I ask you to, do. So what I'm going to do here is I'm going to go ahead and get rid of this because it's down here, right? I'm going to scatter plot the numbers in column B and C. I'm going to click on Insert, Scatter Plot. Now, this isn't the actual scatter plot. I'm going to move it up to the top here. And I'm going to click on a dot here. That'll allow me to move this blue box to the yield. And again, this is the scatter plot for all the observations. And then what I'm doing, this is for all 48. This is for n equal 48. I'm going to make 2 copies of this. The first copy is going to be n equal 18 because there's 18 greenhouses in the southern rule. So I'm going to put southern greenhouses. I'll put n equal 18 below it. Click on this white area. I'm going to grab one of these corners. I'm going to move it up until we get to 18. So this is the scatter plot for the southern greenhouses. And then I'm going to do the same thing for the northern greenhouses. How many northern greenhouses are there? 30. Then I'll click on the white area here. And I'm going to move this over here. I'll bring this corner down to 19. So this is going to scatter plot 19 to 48. I'm going to show you a really cool trick here. I'm going to copy this. I'm going to paste it on top of this. And you can see the orange dots correspond to the southern greenhouses, and the blue dots correspond to the northern greenhouses. Notice the southern greenhouses, the orange squares, are higher. So for a given level of CO2, they generally are associated with higher levels of yield. Why? Because the growing season is longer in the south than it's in the north. The days are going to be on average longer in southern climates than in northern climates. And we're holding everything else constant, right? Also notice that we have a couple of southern greenhouses where the yields make the relationship look less linear. It kind of looks like it's curving, right? So maybe there's something weird going on in these 2 greenhouses. Whereas when you look at the blue dots, it's a line, right? I can click on the blue dots and I can insert a trend line. This is the equation for the blue dots. I can do the same thing for the orange dots, the southern greenhouses. I'll make this bold and I'll make the font dark orange. And I'll make this bold and I'll make these dots blue. Right click on this so we can see it better. I'm going to change the minimum value to 15,000. I'm going to change the minimum value to 200. And hopefully we can see it better. Then I'll stretch this. Now, if you look at the blue dots around this black line, they lie closer to the black line. If you look at the orange dots around this black line, they're spread out more. And there also seems to be like something nonlinear in the data. Maybe there's a lot of variation within them. Maybe the soil is different. Maybe some of these greenhouses are located in heavily dense forested areas. Or maybe some of them are located closer to cities or highways. And so that's creating some noise in our data. And you can see why this line is steeper. This dot here really pulls on the line to rotate it up, whereas this data looks very linear related. Now, one thing that we need to be worried about here, if you go back to this graph, if you look at this graph, it's nonlinear, is it? But you got to remember that the range of our data here goes from just under 250 to 550. So this graph here, we got 250 to 550. From 250 to 550, this part of the curve is very linear. Because CO2 can be very expensive to purchase for a greenhouse, greenhouses are probably going to maintain CO2 levels on the lower end. They're not going to have CO2 levels up here. If your CO2 level is being maintained between 2,000 and 1,600, you're really getting the same effect as if it was between 250 and 600. So there's no reason to spend the kind of money you would need to get CO2 levels this high. So we're going to be on the left linear side of this parabola. If the CO2 levels went out to 1,600 just instead of like 550, we might have to modify the model by doing this. We'd have to include CO2 squared. And the way we would model it is we'd do a multiple regression and we would square these values here. And we'd include both of these as x's. Now that we don't have to do that here. Why? Because again, the parabola is pretty linear in the range of CO2 that we're looking at. Okay, so I'm going to go ahead and delete that column.